nutrient sensing and RGC survival in glaucoma. Glaucoma is a major cause of irreversible blindness worldwide, involving damage and loss of the eye's retinal ganglion cells, RGCs, and their axons. These cells send visual signals from the eye to the brain, so their health is vital for vision. Current glaucoma treatments lower eye pressure, but many patients still lose vision, highlighting the need for neuroprotective strategies that directly support RGCs. Emerging research shows that how RGCs sense and use nutrients like amino acids can influence their survival under stress. In particular, the mechanistic target of rapamycin, mTOR pathway, and autophagy, a cell's recycling program, play key roles in RGC health. This article explores how amino acids, especially leucine, a building block of protein, affect mTOR and autophagy in RGCs under glaucomatous stress, and how we might test dietary interventions to help protect vision. We also discuss how to measure both structural, OCT imaging, and functional, PERG-VEP, outcomes alongside blood CSF biomarkers of nutrient signaling, and consider the balance between growth signals and protein cleanup in cells. mTOR and autophagy, balancing growth versus cleanup. Cells constantly balance between building up structures and recycling damaged parts. mTOR is a master growth sensor. When nutrients are abundant, mTOR turns on protein production and cell growth. Under those conditions, mTOR suppresses autophagy, the cell's recycling bin that breaks down damaged components. In contrast, when nutrients or energy are low or stress is high, mTOR activity falls and autophagy is activated, helping cells survive by cleaning up waste and providing raw materials for energy. In healthy neurons, a basal level of autophagy is important to remove misfolded proteins and worn-out mitochondria. RGCs are especially vulnerable to damage because they are long-lived nerve cells that cannot dilute waste by dividing. Studies show that autophagy protects RGCs under stress. For example, one landmark study found that blocking mTOR with the drug rapamycin, which boosts autophagy, helped RGCs survive after optic nerve injury. In glaucoma models, enhancing autophagy was generally neuroprotective. As Boya and colleagues explain, stressed RGCs use autophagy to reduce oxidative damage and recycle nutrients, which can prolong cell survival. In short, keeping autophagy active helps RGCs stay healthy, especially under the chronic stress of glaucoma. However, too much autophagy or mistimed autophagy can also be harmful, so the balance is delicate. Excessive mTOR inhibition, overactivating autophagy, could have broad effects. The interplay between mTOR and autophagy in RGCs is complex. For example, shutting off mTOR can reduce protein synthesis needed for repair, while hyperactive mTOR from too many nutrients can starve the recycling system. This balance must be managed carefully in any intervention. Leucine and amino acid signaling. Amino acids are not just building blocks of proteins, they are also key regulators of cell metabolism. Leucine is one of the 3 branched-chain amino acids, BCAAs, along with isoleucine and valine. Leucine is a potent activator of mTORC1, the nutrient-sensing complex of mTOR. When cells detect leucine, a cascade involving sensors like cestrin-2 and RAG-GTpases drives mTORC1 to the lysosome and turns it on. This signals that nutrients and energy are available, so the cell ramps up protein synthesis and growth processes. In contrast, low amino acid levels, as in starvation, inactivate mTOR1, lifting the brakes on autophagy. In effect, cells eat themselves to recycle amino acids into energy. A recent molecular study showed that leucine-derived acetyl-CoA leads to modification of the mTORC1 component raptor, which switches on mTORC1 and turns off autophagy. In short, when leucine is present, the cell treats it like a signal to grow rather than recycle. Leucine also influences other nutrient sensors. For example, cell energy stress activates AMPK, AMP-activated protein kinase, which turns off mTOR and conserves energy. High leucine and other nutrients can blunt AMPK and reactivate mTOR. Moreover, insulin, another anabolic signal, strongly activates mTORC1 tau through the PI3K ACT pathway. In RGCs, insulin receptors are abundant, and insulin signaling promotes cell survival and regeneration. Intriguingly, intranasal insulin is being tested as a glaucoma treatment. Thus, RGCs respond to a network of nutrient signals. Amino acids like leucine, hormones like insulin, and stress signals like AMPK all converge on mTOR to determine cell fate. Nutrient sensing in glaucoma. Preclinical evidence. Recent preclinical studies have begun linking nutrient pathways to glaucoma. In animal models of ocular hypertension or genetic glaucoma, RGCs show signs of failing energy metabolism. For example, elevated eye pressure triggers AMPK hyperactivation, a starved stress state, and a drop in ATP levels in RGCs. Persistently active AMPK shuts down high-energy processes. RGCs retract their dendrites, lose synapses, and their axonal transport of mitochondria and proteins stalls. One key study found that inhibiting AMPK under these conditions restored mTOR activity and protected the RGC structure and function. In short, keeping mTOR on via nutrient signals can rescue stressed RGCs. A number of experiments have looked at directly giving nutrients to boost RGC survival. Hasegawa and colleagues showed that supplementing retinal cells or animals with BCAAs, especially leucine, greatly improved energy production and prevented cell death. In cultured cells under stress, adding a mixture of leucine, isoleucine, and valine raised ATP levels and reduced cell loss, whereas simply adding sugar did not. In mouse models of inherited retinal degeneration, including glaucoma-like RGC loss, daily BCAA supplements started even in late-stage disease, significantly slowed RGC death. In one glaucoma model, glassed knockout mice, which lose RGCs over time, mice given BCAA in their drinking water retained thicker nerve fiber layers and more surviving RGCs at 1 year of age. These treated mice had on average 15% more RGCs and a larger optic nerve area than untreated controls. In other words, BCAA, rich in leucine, treatment protected RGC structure in a glaucoma model. Biochemically, the BCAA-treated mice showed less stress in their retinas. Markers of endoplasmic reticulum stress, like CHOP, were reduced, and levels of phosphorylated S6 kinase, a readout of active mTOR1, were higher in treated eyes. In fact, BCAA-treated RGCs tended to restore mTOR activity toward normal. Together, these data suggest that extra dietary leucine helps RGCs survive by feeding energy metabolism and reactivating mTOR-driven growth programs while easing stress responses. On the other hand, some studies warn that too much mTOR signaling can be harmful if it blocks needed cleanup. In diabetic retinopathy models, excessive BCAAs actually worsened inflammation in retina support cells via overactive mTOR. This highlights a potential trade-off. While leucine can feed RGCs, chronically high mTOR may cause buildup of toxic proteins if autophagy is suppressed too much. For example, in other neurodegenerative diseases, like Parkinson's and Alzheimer's, imbalanced nutrient signaling is thought to play a role. Overall, the preclinical evidence indicates nutrient sensing is critical in optical nerve health. Boosting anabolic signals, mTOR, can rescue stressed neurons, but must be balanced against the need for proteostasis. Proposed leucine amino acid interventions. Based on these findings, one potential strategy is to test controlled doses of leucine or BCAAs in glaucoma patients to support RGC survival. Animal experiments used quite high doses. In mice, about 1.5 grams of BCAAs per kg of body weight per day in drinking water was effective. For a human, an equivalent dose by body weight scaling would translate to several grams of leucine each day. A typical BCAA supplement pill or protein-rich meal contains on the order of 1 to 5 grams of leucine. Dose-ranging trials could start at modest levels, e.g. supplemental 2 to 4 grams of leucine daily, and adjust upward carefully, monitoring for effect. Because excessive mTOR activation may have downsides, such trials should proceed cautiously. For instance, giving high-protein supplements chronically could tax the kidneys or tip the balance away from autophagy. Therefore, safety and biomarkers must be tracked. In liver disease patients, BCAA supplements, often in a 2:1 ratio of leucine, isoleucine, valine, have been given daily without severe toxicity. Similar formulas, like the Levac mix used in experiments, could be repurposed. One design could compare a low-dose group, e.g. 1 to 2 gram leucine daily, versus a higher dose group, 5 to 10 gram leucine versus placebo, over several months. Throughout, we would measure nutritional intake and blood levels of amino acids to confirm dosing. It may also be worthwhile to assay mTOR activity indirectly. For example, measuring levels of phosphorylated S6 kinase, PS6K, or other mTOR targets in peripheral blood mononuclear cells, PBMCs, might indicate systemic mTOR activation, though this is indirect. More directly, newer assays could attempt to measure amino acid sensing signals in serum or CSF, if available. For instance, variations in insulin, IGF-1, or even cerebrospinal leucine could serve as biomarkers of the intervention's effect. Combining structural and functional endpoints. In order to evaluate whether amino acid supplements are helping RGCs, multiple types of tests would be combined. Optical coherence tomography, OCT. Scans can measure the thickness of the retinal nerve fiber layer and the ganglion cell layer. Increases or slower thinning on OCT over time would indicate structural preservation of RGCs. In the mouse study above, treated eyes had visibly thicker nerve fiber layers on histology. In patients, OCT can serve a similar purpose. Functional tests like pattern electroretinography, PERG, and visual evoked potential, VEP, would assess RGC function. PERG measures the electrical response of RGCs to visual patterns, and VEP measures the signal reaching the visual cortex. Together, they can detect subtle improvements in retinal function that precede field loss. For example, if leucine supplementation truly protects RGCs, one might see a stabilized or improved PERG waveform amplitude or shorter VP latency compared to controls. Indeed, PERG and VP are being used in clinical trials to gauge neuroprotective strategies. Finally, blood or CSF biomarkers would help link nutrient levels with outcomes. One could build a panel including plasma leucine, isoleucine, valine, the BCAAs, as well as related metabolites, glutamine, glutamate, and systemic signals like insulin or IGF-1. Measuring changes in these nutrients before and after supplementation would confirm uptake. In parallel, stress markers, like neurofilament light chain or glial fibrillary acidic protein in blood CSF, and metabolic markers, NAD plus NADH ratio, ATP levels, could provide additional evidence for improved cellular health. Combining these structural, OCT functional, per VEP, and biomarker data would give a comprehensive picture of an intervention's effect on RGC degeneration. Trade-offs: growth versus proteostasis. A key consideration is the balance between anabolic signaling, growth, and proteostasis, protein homeostasis. Activating mTOR with leucine can boost cell energy and growth, but it inherently suppresses autophagy. Over the long term, this could allow damaged proteins or organelles to accumulate in RGCs. Indeed, one of the touted harms of hyperactive mTOR in aging is that it can drive plaque formation, as seen in Alzheimer's models, by reducing autophagic cleanup. In RGCs, diminished autophagy could theoretically accelerate neurodegeneration if cellular debris is not cleared. Therefore, any nutrient-based therapy must consider this trade-off. One idea is to use intermittent or cyclic dosing, for example, days of leucine supplementation followed by days of autophagy recovery to keep the system balanced. Another approach is to combine leucine with agents that selectively support autophagy, for instance, low-dose rapamycin pulses or AMPK activators, to mitigate buildup. While speculative, current knowledge suggests moderate mTOR activation to support RGC repair and energy might be most beneficial, rather than continuous maximal stimulation. Ultimately, personalized monitoring will be key. If a patient on high-dose amino acids shows signs of impaired clearance, rising markers of protein misfolding, for example, the regimen could be adjusted. The goal is to harness the protective effects of nutrients without tipping the scales toward detrimental protein aggregation. Conclusion: retinal ganglion cell degeneration in glaucoma involves metabolic stress and energy failure. Preclinical evidence points to nutrient pathways, particularly the mTOR autophagy balance controlled by amino acids like leucine, as a modulatable factor in RGC survival. Studies in mice show that boosting blood amino acids, BCAAs, can preserve RGC structure and function, likely by increasing ATP production and reactivating growth signals. Translating this to human treatment will require careful dose finding and monitoring. Clinical trials could test leucine or BCAA supplements, tracking OCT images of nerve fiber thickness and PERG VEP responses as outcome alongside blood levels of nutrients and mTOR markers. This nutritional approach is not a substitute for standard glaucoma care, but it offers a complementary strategy. By feeding RGCs the nutrients they need, we may strengthen their resilience under disease stress. Still, we must ensure that promoting growth signals does not compromise the cell's cleanup systems, a trade-off between anabolism and proteostasis. With well-designed studies combining imaging, electrophysiology, and biochemical panels, researchers can clarify the optimal amino acid dosing and its real impact on preventing vision loss. In the meantime, maintaining a balanced diet with adequate protein and especially essential amino acids remains a reasonable general recommendation for patients concerned about vision and health. All links to sources are available in the text version of this article. You can find the full article at visualfieldtest.com. Thanks for listening. To check your visual field, click the link at the bottom of this article or visit visualfieldtest.com.